Hello student, my name is Assistant Professor Vaishali Sonawane. Presently I am working in MVP's S.V.K.T College, Deolali camp in the Department of Microbiology. Welcome to my lecture series on bacterial pathogen. Today we are going to discuss on Vibrio cholera. This topic is meant for TY-BSc Microbiology student semester 1. So let's begin our lecture with a brief recap of what we have discussed in our last lecture. So in our last lecture, we have discussed about the pathogen, bacterial pathogen Salmonella typhi. Under that, we have discussed about the laboratory diagnosis of Salmonella typhi, followed up by treatment and prevention and control of the infection that is caused by Salmonella. Today's session agenda is to study morphological characteristics of Vibrio cholera, classification of Vibrio cholera, cultural characteristics of Vibrio cholera, followed up by biochemical characteristics of Vibrio cholera. As far as learning objective for today's lecture is concerned, the main learning objective of today's lecture is to study the morphological characteristics of Vibrio cholerae, followed up by cultural and biochemical characteristics of Vibrio cholerae. Now, when we talk about Vibrios, the word Vibrio is derived from the word vibratory movement of that particular organism. When we talk about Vibrio, they show a typical vibratory motility that is called as your darting motility, and the characteristic name Vibrio is derived from the vibratory motility that is performed by this particular organism. When we talk about Vibrio, the genus Vibrio, it consists of near about 33 species of curved bacilli. If you see the morphology of Vibrios, they are comma-shaped organism, and among these 33 species, 12 species are found to be associated with the bacterial pathogen. And the very most important pathogen of this genus Vibrio is Vibrio cholera, which is associated with a well-known disease, that is cholera. This cholera bacilla was first isolated in 1883 by Koch in cholera patient from Egypt. Now, when we talk about cholera, cholera is nothing but it is an acute diarrheal disease. And acute diarrheal disease in the sense, this particular diarrheal disease has an onset of diarrhea within fraction of seconds. Right? And in this particular acute diarrheal infection, what happens is that there is a constant 2 to 3 times diarrhea generally occurs per hour in that particular patient. Now, when we talk about acute diarrheal infection or the diarrhea which is associated with cholera, it is generally untreated if it leads to death of the person if it is untreated. Research has estimated that near about 1.3 to 4 million cases of cholera occurs every year, and among all these cases of cholera, 21,000 to 1,43,000 of death generally occurs because of the cholera. Most of the infections, in most of the patients, this infection remains, it has a mild symptoms or it is asymptomatic. But when we talk about the severe infections, when there is ample amount of vibrios entering into the gastrointestinal tract of the patient, that generally requires your intravenous fluid therapy along with the treatment of antibiotics. Now, how this particular infection can be controlled? Now, if there is a provision of safe drinking water, if there is a proper sanitation condition, if it is maintained, then it is very easy to control such type of waterborne infection. Not only cholera, but any other type of waterborne infections can be cured by maintaining a personal hygiene and by maintaining a proper sanitary conditions. When we talk about cholera, oral cholera vaccine is also available, which along with the sanitation or along with proper hygienic condition can help to avoid the occurrence of infection of cholera. In 2070, a global roadmap is a strategy target to reduce cholera. This particular ending cholera strategy was put forward, which has an aim of ending or to reducing the cases of cholera till 90% till year 2030. Now let's begin with this particular pathogen, Vibrio cholera. First we'll discuss about the morphological characteristics of Vibrio cholerae. Vibrio is nothing but it is a gram-negative bacilli, gram-negative bacteria. This particular organism is basically a curve shape. If you see the morphology of this organism, it is a slight curve in shape, curve rod you can say, and this has a tendency to undergo pleomorphism. Now what is pleomorphism? Pleomorphism is the phenomena of changing the shape of that particular bacteria. If you incubate this organism for longer period of time or because of constant subculturing, there is a tendency that this organism changes its shape from curve to rod. So this organism can exist in these two morphological shape. One is curved one and another one is your rod. Even in fresh culture, you can observe a spiral shape organism. This spiral shape or S shape, this arrangement generally occurs because of lying of two or more vibrios in a row. So that gives a shape of spiral or S shaped or curve shaped. Now this particular organism is motile one. There is a characteristic motility which is performed by this organism, and when we talk about motility, flagella is always associated with a motility. So if an organism is motile, surely it is going to have a flagella. Now when we talk about Vibrio, Vibrio has a single polar flagella. It has a single flagella at its one end, and along with flagella it also has a pili. Now, since we are studying different pathogen in this particular lecture series, we have seen that pili plays an important role as far as attachment of this organism is concerned, because whenever any microorganism enters into the body of a host, the very first thing is the establishment of that particular pathogen. The organism need to get established in the body of host, and pili plays an important role as far as attachment of this organism is concerned, because attachment, if the organism is able to attach to mucous cell lining, only then and then it is going to multiply, it is going to increase its number, and when it is going to increase its number, then it is going to cause a severe type of infection. So pili is also associated with your Vibrio cholera. As I told you that the organism is motile and a characteristic motility is performed by this organism. That characteristic motility is called as darting motility. The name darting motility is given because it resembles with that of the movement of dart. This organism is non-sporulating ones. Even though it is a rod, it doesn't form any type of spore, and it is non-capsulated. So capsule doesn't play any role in the pathogenesis of this organism. It lacks capsule. Let's see the morphology of Vibrio. In this particular image, you are able to observe two images. One is the one which indicates the morphology of Vibrio, and another one is associated with gram staining of Vibrio. So first, let's see the morphology of Vibrio. Now, as we have already discussed in our previous slide, that Vibrio is curve-shaped bacteria and it is associated with a single polar flagella. So in this particular image, you can see this a particular curve is there, and this is a polar flagella. You can see the single polar flagella is present in case of Vibrio. So this is a typical morphological characteristic of Vibrio. Moving on to the next image is the gram staining of Vibrio cholera. Now even in the gram staining, as we have seen that the organism is gram negative in nature, the organism stains pink after performing gram staining, and even in this particular image you will be able to see a curve. See, there is a curve in the in this particular image. The organism appears curve shaped and shape. So this is the morphological characteristics of Vibrio cholera. Now let's move on to the classification of Vibrio cholera. Now when we talk about classification of Vibrio cholera, O polysaccharide, O somatic antigen, which is present on the cell wall of Vibrio cholera, plays important role in the classification of Vibrios. It is divided into many serotypes. This particular classification on the basis of O polysaccharide was given in 1935 by Gardner and Venkatraman. In this particular classification, there are over 140 different serogroups of Vibrio were found, and they were designated as O1 to O140. And among all these O1 to O40, all of them are morphologically they possess the same characteristic as that of Vibrio, but only O1 and O139 serogroups are associated with the production of toxin. See, when we talk about cholera is caused not because of the presence of bacteria, but it is caused because of the toxin that is produced by your Vibrio. And this particular toxin production is generally present in O1 and O139 species, and therefore they are associated with cholera in case of human beings. Now, this O1 serotype, it is further divided into three different serotypes: Inaba, Hikojima and Ogawa. These are the three different serotypes of O1 Vibrios. This Inaba, Ogawa and Hokijima, these three have different O type serotype on their cell surface. Inaba and Ogawa, these are generally agglutinated by their own antisera. So if there is a strain of Inaba and if you want to detect it, you will require a same antisera. Similar is in case of Ogawa. But when we talk about of Hikojima, Hikojima is an intermediate between that of Inaba and Ogawa, and Hikojima is agglutinated by antisera of both of these organisms. So in this particular table, you will be able to see the major O factor which is present in Inaba, Ogawa and Hokijima, and the agglutination. Now when we talk about the serotype Ogawa, the major factor which is present is A and B factor, and it is agglutinated by only its own antisera. When we talk about Inaba, it has A and C as a major O factor, and it is again similar to that of Ogawa. It is agglutinated by the antisera of Inaba, and the last one is Hikojima. Hikojima, the major factor which is present is A, B and C. So it possesses all these three factors, and because of this particular reason, the organism is agglutinated by antisera of Ogawa as well as Inaba. It is said that the organism has a tendency to change from Inaba to Ogawa, and the Hikojima is the intermediate between Inaba and Ogawa, and therefore the Hikojima has a tendency to get agglutinated by the antisera of both Ogawa as well as Inaba. Now, when we talk about further classification, the classification which we have seen is the one which is based on serogroups, serotypes. Vibrio O1 is again divided into based on the morphological characteristics and phenotypic properties into biotypes. It is divided as classical and even TOR Vibrios. Okay. Now this even TOR Vibrios, all the pandemics, now we all are aware that there are near about 7 pandemics since 1890 which has occurred of cholera, and in that the first 6 is caused because of the classical Vibrios, but the last pandemic is found to be caused because of even TOR Vibrios. So this is a flow chart which is representing a classification of your Vibrios. See, the Vibrios are categorized as group A and group B. Group A Vibrios, they has a common H antigen, and they have O antigen is variable. And when we talk about B group, they are morphologically similar to that of Vibrio, but these are biochemically as well as antigenically heterogeneous group. When we talk about group A, on the basis of serogroups, O group is categorized as your the organism which are agglutinated by antisera of O1, and the organisms of or the Vibrios which are not agglutinated by your O1 antisera. The Vibrios which are not agglutinated by your O1 antisera is also called as NAG Vibrio, and that NAG Vibrio are found to be non-pathogenic one. And O1 Vibrios are further on the basis of their morphological and phenotypic properties, they can be classified as your classical and even TOR Vibrio, and they can further classified into 3 serotypes: Ogawa, Inaba and Hikojima. This the last pandemic which was caused by even TOR Vibrios, this particular Vibrio was isolated. The name tor was given in due respect to the place from where the organism is isolated. It was isolated from the Hajj pilgrims of the pilgrims which died because of the dysentery, and the organism which was isolated was found to be the Vibrio, and but it has a distinct characteristic. It is slightly different than on the genetic level as compared to that of the classical one, and therefore this particular the station was Tor quarantine station, and because of that the name is given even Tor vibrios to this category of organisms. Next we are going to move on to the cultural characteristics of this particular organism, how you are going to grow this organism if you want. Now when we talk about Vibrios, Vibrios are facultative anaerobic in nature. So they has a tendency to grow under aerobic condition as well as anaerobic condition, but they generally prefer aerobic condition as far as their growth is concerned. The temperature ranges from 16 to 40 degrees centigrade, and 37 degrees centigrade is an optimum temperature for their growth. Now here we are talking about Vibrio cholera. Now Vibrio cholera, since it is a human pathogen, so the temperature is near about 37 degrees centigrade, which is an optimum temperature required for growth of this organism. As far as pH is concerned, the organism requires an alkaline conditions for its growth as well as development. 8.2 is the optimum pH which is required by this particular organism, but it can grow within the range of 6 to 9.4 pH. This Vibrios, generally the genus Vibrio is the halophilic one. They generally require a higher concentration of salt for their growth and development. But when we talk about Vibrio cholera requires a less concentration of salt as compared to that of other Vibrios. So it basically requires 0.5 to 1% of salt concentration, and its growth is inhibited about 6% of NaCl. Now when you are going to culture this organism on nutrient agar, the pH should be maintained within the range where the organism generally grows. So on that, on nutrient agar, the organism produces a moist, translucent, round and bluish tinge colored colony which has a distinctive odor. If you talk about McConkey's agar, if we are going to cultivate this organism on McConkey's agar, this organism generally in some references it is given that the organism is lactose negative or the organism doesn't produce pink color colonies on McConkey's agar. But when we talk about Vibrio cholera, this organism is a late lag fermenter. Now what is late lag fermenter? Late lag fermentation is the one, or late lag fermenter organisms are the one which produces pink color colonies on McConkey's agar after a prolonged incubation. Means if you are going to culture it today, after 24 hours you won't be getting a pink color colony, but if you prolong the incubation for 48 hours or for 72 hours, then the organism will surely show you pink color colony. So your Vibrio cholera is a late lag fermenter. Similarly, if we cultivate this organism on blood agar, the organism results in alpha hemolysis, which is followed up by beta hemolysis. Alpha hemolysis is called as your partial hemolysis. So there is a partial hemolysis of RBC, there is not complete hemolysis. But when we talk about beta hemolysis is the one in which there is a complete hemolysis of your RBC. This thing can be visualized by the zone color which is formed around the colonies of your Vibrio. Now we'll see the growth of Vibrios on selective media. Now basically the selective media which is used for cultivation of Vibrio is TCBS agar. This is an image which is showing you the growth of Vibrios on TCBS agar. On TCBS agar, Vibrio result in formation of yellow colored colony, and this yellow color is formed because of the bromothiamyl blue indicator which is present in the media, which under alkaline conditions changes its color to yellow, and because of this the colonies appear yellow in color. Similarly, this next image is the image of Vibrio on blood agar. You can see there is two zones of hemolysis. The first one is the blur zone which is followed up by a clear zone. So the blur zone is of your alpha hemolysis and the clear zone indicates the beta hemolysis. So there is a typical hemolysis which is carried out by this organism on blood agar. So even this thing plays important role as far as diagnosis of this organism is concerned. So merely by looking on the plate, you can able to identify that the organism might belong to the Vibrios. Now, along with your selective media, the organism also has a tendency to hydrolyze gelatin. The organism is gelatinase positive. It has a tendency to produce gelatinase enzyme, which also helps you to identify this particular organism. As far as gelatin liquefaction is concerned, a typical funnel shape liquefaction is observed at 33 degrees centigrade. On peptone water, a thin pellicle growth is observed. If you prolong the incubation, a powdery deposit of Vibrio generally occurs at the base of the tube. Now, when we talk about Vibrio, now throughout our lecture or in the beginning of lecture, we have seen that this particular organism is quite different from that of other pathogens, and this organism requires different conditions for its growth and for its survival. Now, because of this particular reason, a typical holding media or transport media is designed, which generally helps to carry the organism from one place to another. Like from, if there is some epidemic at one particular rural area, their laboratories are not available and you need to send the sample to some other laboratory. In that case, you can't send the stool sample or vomit sample. In that case, you require a holding media or a transport media. So for Vibrios, a holding media and transport medias are generally designed, which are designed in such a way which only helps the organism to survive. It doesn't favors the growth or multiplication of this particular organism. So in case of Vibrio, the holding or transport media is VR media. VR media is Venkatraman Ramakrishnan media. This Venkatraman Ramakrishnan media is the name is given due respect to the scientist who designed this particular media. Then Carrie Blair's media, and even the sea water, normal sea water, which is properly autoclaved, can be used as a transport media for Vibrios. And when we talk about plating media, means when you need to grow the organism or when you need to transfer this organism from transport media, you require a plating media. And the plating media which is used for Vibrio is alkaline bile salt agar and TCBS agar. Now, when we talk about biochemical characteristics of Vibrio, the organism is gelatinase positive. In this particular image, you can see that after inoculation of this organism in a gelatin media, after incubation at 37 degrees centigrade, you can see the liquefaction of the gelatin. So gelatin is hydrolyzed by the organism and it is broken down, which loses the property of solidification of this gelatin, and there is a liquefaction, and this particular whole liquefaction is imparted by the production of gelatinase enzyme by this particular organism. So your Vibrio cholera is a gelatinase positive. Then the second character is that the organism is oxidase positive. The organism has a tendency to produce oxidase enzyme and it result in formation of purple color when the colony reacts with your oxidase reagent. Along with that, the organism is also a catalase positive. So when a culture of Vibrio cholera, when it is mixed with H2O2 solution, then there is an effervescence of oxygen and it indicates that the test is positive. So the organism is your catalase positive. All these three tests, these are the common tests and easily available tests which can be used for laboratory diagnosis of your Vibrio cholera. So the organism is gelatinase positive, oxidase positive, as well as catalase positive. Okay. Now, among your gelatinase, catalase, oxidase, along with that, the organism is your has a tendency to reduce nitrate to nitrite along with production of indole when it is grown in peptone broth. Right. Now both these reaction, that is production of indole and reduction of nitrate to nitrite, both of these reaction contributes to a reaction called as Cholera Red reaction. Now in this particular reaction, when the organism is grown into a media, peptone broth, it result in production of indole, and along with that it result in conversion of nitrate to nitrite, reduction of nitrate to nitrite, and after incubation, when H2SO4 is added, few drop of H2SO4 is added to the media, it result in formation of red color. So in this particular image you will be able to observe the red color that is formed. This red color is formed because of an complex which is formed, that is nitrose indole complex, that is formed because of the reduction of nitrate to nitrite and because of indole production. So Cholera Red reaction is very important biochemical characteristic of the organism which can be used for identification of cholera from sample. Now let's see what we have learned in this particular lecture. So in today's lecture we discussed about Vibrio cholera. As far as Vibrio cholera is concerned, we have discussed the general characteristic of genus Vibrio, followed up by classification of Vibrio, morphological characteristics of Vibrio cholera. Then we have gone through cultural characteristics of Vibrio cholera, and lastly we have discussed the biochemical characteristics of Vibrio cholera. The cultural characteristics as well as biochemical characteristic plays important role as far as laboratory diagnosis of this organism is concerned, and therefore it is necessary that you should be aware of the basic cultural characteristics and biochemical characteristics for identification of the pathogen. Now there is a small assignment for all of you. Very first thing is that you are supposed to write a note on holding media that is used for Vibrio. So you are supposed to write down the names of the media, you are supposed to write down the composition of the media and the condition of the media. Then second thing, you are supposed to write what is the Cholera Red reaction. So in this particular question you are supposed to write details of Cholera Red reaction what we have discussed in our lecture. So let's have a brief recap of our next lecture. So in next lecture we are going to discuss on the determinants of pathogenicity of Vibrio cholera. So what are the various factors or virulence factor which plays important role in the pathogenicity of Vibrio cholera. Then we are going to see the pathogenicity, how Vibrio cholera enters in the body of a host, how it grows, how it multiplies, how it produces toxin, and basically how it causes the infection known as cholera, followed up by the symptoms. Then after pathogenicity we are going to see the symptoms that are associated with cholera. Then we are going to see the laboratory diagnosis. So one symptom is there, which type of sample you are going to use for identification of this organism, and how you are going to proceed for that particular sample, that we are going to see under laboratory diagnosis. And then we are going to see the treatment and control of this particular infection. And lastly we will have a brief story of cholera, which is going to give you an overview of whole two lectures of Vibrio cholera. So thank you.